It's a good morning. It's a pleasure to have Dr. Philip Kuehl of the Lovelace Biomedical Research Institute with us today. And he'll be participating in our Bench-to-Bedside seminar series to present his work on translational inhalable or inhalation drug delivery and the development of inhaled biologics. Having been a kid in Brooklyn, and seeing people inhaling drugs on every street corner, I can probably teach Dr. Kuehl a number of things. But and I was a kid, so he had to get a Ph.D. to learn this stuff. And so he got his Ph.D. in pharmaceutical sciences at the University of Arizona in Tucson, Arizona. Since 2007, Dr. Kuehl has been at the Lovelace, rising from associate research scientist to the position of Scientific Core Laboratories. And he's the director there, where he oversees the chemistry, the aerosol, microbiology, virology, telemetry, and immunology laboratories at Lovelace. He's also a research associate professor at the College of Pharmacy at the University of New Mexico in Albuquerque. And Dr. Kuehl research focuses on the development and characterization of inhalation products. And he's really looking for new formulations that will increase the delivery to targeted sites while minimizing off-site delivery and off-site toxic effects. He has tons of publications in that field, and I told you I'd be short.
That was perfect. Thank you.
I am perfect.
So I should start out with one quick joke then, as we're talking about inhaling things. I've actually been a part of about 5 or 6 different clinical studies where we're looking at deposition of where things go in the lung. And I did these, I don't know, 7 or 8 years ago, so I was even more young and impressionable then. And I had one patient actually use an inhaler correctly. They inhaled nicely, they did a great breath hold, and then exhaled. I'm like, where'd you learn how to do that? Who's your pulmonologist? Who was your physician? Was it a pharmacist that taught you that? The gentleman looks at me and goes, well, when I was in college, I smoked a lot of weed. And I learned that I got higher faster if I held my breath. I'm like, you know what? I'm done. I don't want to interact with patients anymore. So today, I'm going to give a little bit of a different style of seminar because I want to do a couple of things here. One of the main things I want to do is show you what things are possible and also give you a little bit of breadth of what the heck this company, Lovelace Biomedical, is, that we're all joined together now and kind of have this opportunity to do cool stuff, what we can do and what our strengths are so that you can see using some case studies of what we can do. So if you can't tell, I'm pretty chill and pretty laid back. Ask questions. If there's something going on in the middle, I'd actually rather take the last 5 or 10 slides and just say, hey, here's another manuscript. I'll send that to you if there's good conversation. So do what is right on that. I want to give you a background, like I said, of what Lovelace is and what translational research is for Lovelace, what we mean by that, because everyone says those things and they can mean somewhat different things. I want to talk a little bit about some case studies, one of which is pretty topical in the most recent pandemic. Another one is an inhaled biologic that we hope gets some legs and actually goes somewhere. I think this stuff is pretty cool. I think this stuff is a lot of fun to work on. That's why I'm there. That's why I've been there for almost 17 years now. And hopefully, like I said, give you guys a base what we can try to do together. So what is Lovelace Biomedical? Let's start there. We're a private, not-for-profit CRO, contract research organization. Fundamentally, that means we have the ability to use just about any animal model out there you can envision, whether it's for PK, whether it's for toxicology, whether it's for pharmacology models. We have a lot of unique pharmacology models. We're a small company. We are built on science. There are about 7 to 10 folks like me that really the back of that institute is built on doing cool science in and around the area of respiratory, infectious disease, and gene therapy. That's really our best thing. There's all kinds of cool things that we can do, IND-enabling studies and all these acronyms. Don't worry about any of those. Focus on the fact we can use animals in research in areas that most places can't. And we understand we're super lucky at being able to do that. So I went over kind of some of our areas of expertise. We can do all sorts of other things, but usually there's support within using animal models. We do a lot of really cool bioanalytical. We were talking about next-gen sequencing on the way over here. We don't do a lot of that, but we do a ton of PCR and molecular assays. And we have the ability to work in containment with a lot of different viruses and different bacteria strains. Our facility is huge, and we appreciate that that makes us very lucky. We've got like 140 acres, like 370,000 square feet, and give or take about 225 or 250 employees that span the breadth of really supporting drug development. here are some of those core areas. And if you see anything in there that's interesting to you, let's talk. Because probably we've done something in that area in the past. I'm going to give you a couple of quick high-level stuff on each of those areas of expertise. Then let's dig into some actual cool science. So in gene therapy, again, we're a place that's built off the back of scientists. In 2005 or 6, the government put out a call that said, we need a place to do toxicology and pharmacology for gene therapy. One of our scientists did something crazy. And she said, I'll do it. I'll write a response to that. And a lot of us kind of looked at her and said, Janet, what are you doing? She's like, don't worry, I got this. She won the thing. So since 2007, we've been the place that's done all the pharmacology and toxicology for NHLBI, the National Heart Blood Institute, within the government for all investigator-initiated gene therapy. Because of that, we can actually say we've done all these studies and be public about them, not like you can with a lot of pharma companies where you don't get to talk about it early on. You can publicly disclose all of this. This bottom one is the one that I like the most. It's not public which company those are, but there are 4 companies that I know of that are doing inhaled gene therapy programs for cystic fibrosis with an mRNA therapy in phase 1 clinical trials right now. 3 of them, we did the non-clinical work for. So we're pretty fortunate to be able to work on some really cool stuff, to be able to touch this and be able to help companies go from idea to actually get into human trials. This slide, I think, really sums up a lot of our respiratory work. Those are 3 combination product meter dose inhalers that are marketed by AstraZeneca. We were fortunate enough to work with a company named Kuehl Therapeutics that was eventually bought by AstraZeneca. We did all the non-clinical toxicology to get those products on the market. Really good products. Hopefully some of you that are seeing patients actually know of some of those. They're pretty good products. Like I said, we're built on our science reputation. So the fact that we have, I've seen different numbers thrown around, 500, I've seen 1,000 publications. It's a lot in and around respiratory drug delivery. That's what we're known for. What we're able to do with that is to support 10 to a dozen different investigational new drugs to actually get into dose humans every year. And give or take, we've got about 50 pharmacology models that range from asthma, cigarette smoke models of COPD, all the way to cystic fibrosis that we can use. So that's kind of who Lovelace is. Questions on that? Everybody good on that? I would love for there to be stuff that we talk about. I know we have meet some of you later, but if there's things we can work on together, please let's do it. We will work in areas of grants. We work in SBIRs, and we'll do commercial contract work. Anything that's good research, we want to be a part of. Now I want you to talk about some of the things that we can do with some case studies. The first one is on remdesivir. So hopefully, unfortunately, I guess maybe it's the right word. We all probably know what remdesivir is. This was published and it's in Science Translational Medicine. I can send that out later if you guys want to look at it. But where this came from is during the SARS-CoV, the original SARS-CoV pandemic. Lovelace did a lot of work to evaluate what were the animal models that were appropriate to evaluate SARS coronaviruses in terms of efficacy, vaccination, transmission, treatment, all those types of models out there. We therefore knew some of the things to start with that a lot of other places didn't. What I'm about to say sounds bad, and I apologize for that. The African green monkey was something we knew was the best monkey model for SARS coronaviruses. If you look at publications, the publications are heavily weighted to rhesus monkeys. Rhesus monkeys aren't quite as good of a model, but there's a reason for that. The NIH primate centers have rhesus animals. So we were able to say, hey, look, there's different strains of non-human primates out there. Which one is actually the best and what can we use for research? We were able to say, let's use the African green. You will see a bias in publication towards the rhesus for that reason. So what we wanted was we needed obviously to have an ACE2 receptor. We needed to have an animal model that was susceptible to the phenotype of COVID-19. We wanted to make sure that we had some way to quantify an immune response in these animals. And obviously this last one was huge for us. The availability of the animals, the size and the cost. As a place that wants to use animals in research and support research, if we can't get a key resource, whether that's an animal or a reagent, the model's no good. So I can tell you we use Syrian hamsters and African green monkeys as the main models throughout all of SARS-CoV-2. One of the first things we did is we had to characterize the animals, the dose, and the dosing route. And so if you look at some of the early literature around how to challenge an animal with SARS-CoV-2, they were doing, and this, again, I'm going to say things that are tongue-in-cheek, it's all right. They were giving a proverbial viral shower. They were dosing them orally. They were dosing them intranasally. They were dosing them by inhalation. They were doing all of that to the same animal. It turns out that when you shake all of that free and you figure out what's actually causing the disease in the animals, it's the intranasal challenge. And if you think about humans, we're generally, when we're relaxed, like most of us are right now, we're nasal breathers. So it makes sense that in the animals, that disease is entering through the nose and we deliver it through the nose. That gives us the best the best viral replication and the best translatability to humans. And so we stuck with that intranasal challenge. In general, the model is about 6 days. You see a publication that says 7 days, cool, that's good enough. They do it at 5 days, that's the same. What that amounts to is that, not unlike many humans, the non-human primate gets sick, but in general recovers. And so somewhere around day 7, day 8, day 9, their viral titers, wherever you look at them, whether it's in the lungs, whether it's in nasal swabs, or wherever that may be, you're going to see those viral titers almost go back to baseline. Peak of viral load, whether you look at the nasal swabs or the lung, is somewhere around day 2, 3, or day 4, usually around day 3. And you're going to see some of the endpoints I look at here sitting in around that range. The other key to this was to make sure that we were using healthy animals. These studies were done in our B3, so we wanted to make sure that we had animals that when we put them out there and challenge them, everything was good with them. The other thing that I think is probably for a lot of the clinicians, we did in a lot of our natural history studies. Sorry, mine, you guys is where can you see? Okay. When we looked at our natural history studies, we wanted to know, did we actually see anything on x-rays in the lungs of those animals? We didn't. So there's not the pronounced pneumonitis or anything like that that you often see in the human setting or see in some of the human setting. We don't see that in the animal model. So as like any model, this model is a good model, but it's wrong in some way in terms of totally translated humans. And that's one of the differences. So in this particular project that I'm going to show, we were working with Gilead. They're obviously the ones that had remdesivir for Ebola and found out that, hey, this looked in vitro like it was going to be positive for SARS-CoV-2. Let's see what we can do to try to use this for the SARS-CoV-2 pandemic. I'm going to dig into a little bit on the mechanism of action, the science around this, just so you can understand kind of what this is and why there's some other publications that some of the same people I just showed you have put out. So remdesivir is a prodrug. Through a process, it's going to go through the body and end up in these three, and I'm kind of blocking that, maybe step back. It's going to end up in these three different analytes in the body. It's a monophosphate, a diphosphate, and the triphosphate, with the triphosphate being the most efficacious. Both of these analytes, if you go look in the literature, you will see 277 and 524 published on from Gilead to say, hey, maybe one of these is actually more efficacious in terms of final end result in the body. What this does is it's a nucleotide analog, so it stops viral replication. Most of you that see patients have already gone to that next step and say, this is part of when and where remdesivir is incredibly efficacious. So obviously, it has activity. We're just off the bottom there, I apologize, in SARS-CoV-2, MERS, and in Ebola virus, and in SARS-CoV-1 as well. But this graph, I think, actually speaks a lot to the success of remdesivir. And I think I actually have it right here. Is you have to start early in disease. When you have something that is an antiviral that is stopping viral replication, if you can get remdesivir on board early to stop that replication, it will stop disease. It works very, very well. Without saying anything bad about our healthcare system in the US, during the pandemic, the ability to feel as a patient like you were sick with SARS-CoV-2, get into a healthcare provider, get a test, be prescribed remdesivir, and then get an opportunity to go IV, to go in and get an IV delivery 5 days in a row of remdesivir. Our healthcare system back-ended to make sure the remdesivir was not really efficacious in this pandemic. And so I want you to remember that. IV therapy for remdesivir for 10 days is what was out there. So when Gilead came to us, they said this: why don't we deliver this material to the respiratory tract? If we're getting something IV, if we could have somebody breathe on it at home in a nebulizer, would that work better? Would it work the same? They brought that problem statement to us from their CEO level and said, help us. What do we need to do? So their goal was to be able to deliver this outside of the hospital and to be able to see if this had efficacy. At the time, this was in clinicaltrials.gov. They were doing that. Obviously, they abandoned that trial at this point. What I'm not showing you, because I want to talk about cool stuff. In the background, before we could do any of this research, we had to figure out what formulation we're using for remdesivir. We decided to use the actual IV formulation because then they already had their manufacturing, their cold fill, their sterility, all those things were already all done. So if this had worked, instead of taking a brand new formulation, it was literally that vial that could be reconstituted for IV delivery, reconstitute it, pour it in a nebulizer, breathe on the nebulizer. So we did that for efficiency. We also had to prove that that was safe. We had to go through and use animal models of toxicology to say that if you put that material into the lungs, you're not going to have a local toxicological effect that alters your ability to use things. So that was all done and submitted as an IND in parallel to be able to do the studies that I'm going to show you right now. First, before we go to the cartoons, let's start right here. IV infusion of remdesivir is 10 mg/kg. We know that when you're targeting a respiratory disease, if you're able to cope, if you're able to target and deliver that drug, typically you can give a lower dose. We started out with 0.17 and 0.5 mg/kg as our target dose, where we were going to evaluate how much remdesivir there was in the body and in the lungs before we go to efficacy. In order to do that, you have to figure out how to deliver these aerosols to a non-human primate. What I started out with: humans are terrible at taking their medication. Humans are terrible at taking inhalation medication. That's hard to train a monkey to do that. Literally what we do is we put them in a space helmet. We take a commercial nebulizer, the same nebulizer that would have been used had this gone forward. Many of you have used them, I'm sure, in practice. Deliver that to a head dome on a primate, and then have an exhaust system and a buffer system to pull that aerosol away from them so they get a constant stream of aerosol going to them for them to breathe. And we remove all of their CO2 that they're exhaling away from them while making sure that there's no back pressuring like that for the safety of the animals. I appreciate how lucky Lovelace is to have the machine shop to build this stuff, the facilities to be able to have the vacuum, the air, and the support to do all these things. We're super lucky. We get that, but it's literally that, a non-human primate in a chair with a helmet on. So this is probably the graph that's easiest to look at here. The first thing we had to do was pharmacokinetics. We know that dosing a lower dose and giving it to the lungs is going to give a lower dose systemically, but we also believe that we'll be able to give a much lower dose and see similar efficacy. So when we look at the nucleoside analog, when we were looking at that, the IV dose, the high dose inhalation, and the low dose inhalation, that's a log scale. So there's a pretty significant reduction in overall exposure. But now look at this. This is actually a pretty complex necropsy where we actually do a terminal surgical procedure to put the animal under fairly deep anesthesia, open up and remove lung tissue as a part of the terminal necropsy before the animal is actually euthanized, so we can maintain the right version of that monophosphate, diphosphate, and triphosphate version of the analog, and then analyze those with the LCMS. Now, if you look and see at that significant lower dosing, now granted, like I said, this is a tough procedure. Those error bars, if you're going to keep in those error bars, I'm going to smile and say, yeah, they're pretty wide. But you can see the story across the board with a significant reduction in overall dose. We see similar levels of the nucleoside analog in the lungs of those animals. So for us, that's kind of a progression of going from, we did the tox work, now we have the pharmacokinetic study to show the proof of concept. Let's go into where the real expense is. Now let's do an efficacy study inside of the B3. This is what that study was designed to look like. So this is that same graph that I showed you earlier, where you've got challenge on day 1 with intranasal challenge. And we're going to dose those animals 5 days. This is a lot of what we spend our scientific energy on. Remember back to that slide earlier where I said remdesivir was given IV for 10 days? How do we manage? 10 days is what they do in the clinic. But if we do that for 10 days in animals, no matter what happens, those animals are healthy at 10 days. They're going to shed, they're going to be done with this virus. So we have to bring that and adjust the model to make sure that it's translatable. Where we ended up was at 6 days, we bring the animals down, and you can see we did some viral measures of lavage and nasal swabs throughout that time to see if we could find some non-terminal endpoints as well that suggested efficacy from this study. I'm a science nerd, so when I started to see the graphs for these things, I get a little bit excited. So the first one we had is we started to get some bioanalytical results. If you remember, we were about 10-fold different on that earlier graph. Because these animals in the V3, you can't draw a lot of blood from them. If they get sick, it's real difficult to collect those samples. So just imaginarily draw a line, and let's call that 7 because it's a log scale. Let's call that 0.07 to 0.1. We're in the right range. So when we start getting these things, I start to get a little bit excited. This graph is super, super confusing, and I apologize. So let's just kind of keep going. But if you really dig into it, it's showing you that inhaled remdesivir is kind of sort of similar to IV. I'm going to get to a much easier graph to read in a little bit. This is the one I did want to stop at a little. And just really to say that those nasal swabs didn't really show a clear anything relative to it being an indicator for a virus in this model. And the reason I wanted to show that is because of clinical practice and how often nasal swabs are used for a diagnosis of exposure or prevalence of SARS-CoV-2 in COVID-19 patients. And so again, all of these models have issues with them. This was one of them that in this model, the nasal swab may not have been that great. This is the slide I like, and I generally come to this graph because there's always noise around the data. The open blue circles are effectively placebo inhalation. The closed blue circles are placebo or are drug inhalation. Then the red is IV. And you can see this is subgenomic. So this is replicating SARS-CoV-2 in the lung. And you can see that by inhalation, at that significantly lower dose, we're seeing similar or better efficacy from remdesivir. So that's the kind of things we're able to do with some of these models in here. And again, it's unfortunate the situation we were at, that this wasn't something that could roll out clinically because it had the potential to be a really good therapy. Yeah, that's kind of what I just talked about. The other thing I think speaks a lot to some of the teams that worked on this, and I didn't list everyone up there because there was probably the largest co-authorship publication I've ever been on. I think there were 25 or 30 of us, like a book chapter just to get through the authors. But the amount of teamwork and different disciplines that it took to do that, to have the immunologist, to have the veterinary support, to have the virologist, to have the aerosol team, and all those folks to be able to work together to do that was pretty unique and pretty amazing. I will tell you that some of the calls during this time at Gilead, they were stressful. There were times when the reminder was very simple of just remember, 2,500 people died yesterday from SARS-CoV-2. You guys can shave a day off on this. 2,500 people would really appreciate that. So it's a lot of fun to work in these areas, a lot of fun to get to work on a really cool and novel things, but it doesn't go without stress. So it was a good time. Questions on that? Yeah, let's ask questions on remdesivir. Go ahead. We did and we didn't. So the assay we used was just TCID50, was a TCID50 assay. How did we determine what to give to the animals? What we gave was effectively the highest concentration SARS-CoV-2 you could grow in the largest volume you could give to a non-human primate and not have it effectively just rip out of it. We did do studies where we diluted it, and it just made the disease a little bit more mild, and your concentration of virus from your PCR assays were just reduced down. So when you came back to looking at graphs like this, your opportunity to show a delta was smaller. So when there was no lethality in the disease, we just gave maximum feasible dose, and stayed with that so we could see modulation of the disease. Does that answer the question?
You ever determine infectious dose? Because that's always a question.
So that is a great question, and we didn't spend a lot of time on that, especially in the African green model. We did do some of that in the hamsters, but again, how do you define infectious in an animal that doesn't succumb to disease, that doesn't really elicit any terminal or morbid signs, and all you can do is quantify a virus? Effectively, in the animal model, and this is some of the issues with the animal model, that question you're asking is only related to the LLOQ of the PCR or the TCID50 acid. So it's a really great question that animals can't help with. We spent a lot of time dealing with that. In fact, one of the papers I just published was on transmission using an infected hamster and putting naive hamsters downstream and quantifying what their dose was. And we spent a lot of time with the reviewers going over that to finally convince them that it really isn't the lowest infectious dose because they don't really get a strong phenotype of disease. Rather, everything is just related to your assay limits. And that would be something we could work on because we have those great assays, but we don't have access to do a lot of those clinical studies. But great question. And we spent a lot of time digging around on that. Go ahead.
So, you said the model here, monkeys don't get same kind of pneumonitis that hospitalized patients would get.
Correct.
So to me, that raises two questions. One, the way is related to infectious dose, but how do you really know that the viral burden in the lungs they don't get so much pneumonitis is in any way mimicking humans? And then the second question is, well, ventilation, not again, how do you know that if you gave this patient to actually have pneumonitis, you get enough uniform distribution of the aerosol?
So let's take the first question and then remind me when I come back to the second one when I forget what your second question is. You're right. There are all kinds of places to poke holes in this model. And one of them is, I mean, there's lots of them, and it all relates to that more severe disease that we can't reproduce in the animal models. We've tried all kinds of different things like co-infections where we'll actually, compromise the animals and then try to deliver the SARS-CoV-2. We just can't reproduce anything related to pneumonitis. We can't reproduce long COVID. We can't get any of those to recapitulate. And I know that that's a frustration of all kinds of scientists out there to say, well, what can we do to help this patient population? Right now, there aren't any great animal models to be able to do that. So I apologize for that. I'd love to figure that out, but there's not a great way to do it. In terms of the aerosol delivery to be able to treat the disease patient, that goes for every disease that's treated by inhalation, whether it's COPD, hopefully at some point we're treating lung cancer that way, that you have, whether it's remodeling or mucus plugs or anything like that, that is for inflammation that's causing blockages of the airway. The main way to control that is through the right devices and the right aerosol. So in those regions, the biggest thing you can do is control flow rate and breath hold. Give those particles a chance to deposit in that area and to give small aerosol particle size.
Okay, what's particle
size? So the particle size of this was about 2.5, but micron MM80, mass medium aerodynamic diameter. So it's how that particle behaves in the air, normalized way back to spherical particles, unit density, all kinds of other things. If you read literature, the FDA and the literature will tell you between 1 and 5 is what you want to use to get into the lungs. I'll tell you between 1 and 2 or 1 and 3 is much, much better. And as important to that particle size is how much you screen out in the throat through inertial impaction. So if you have a meter dose inhaler, and I love meter dose inhalers, they are a great product for the right people. But if you spray a meter dose inhaler and 40% of your material impacts in the back of the throat, no matter what you're doing, it doesn't matter what your particle size is, you just reduce your dose by 40%. That's why sometimes those spacers are really good, because the spacers allow that aerosol to slow down before you breathe it in. The same thing with some dry powders. Some dry powders allow you to inhale at a very, very high rate. You're impacting 40 and 60% right in the back of your throat. So it doesn't matter what particle size it is because you've screened so much out. So it's those two factors in the humans that matter, the flow rate and the particle size, and in animals, it's just particle size because by putting them on that head dome, you've normalized all that flow rate and inertial impaction. Other questions on the remdesivir stuff?
So if I understood correctly, you gave the highest possible dose to establish an infection in this model because they do not naturally get infected or they do not. So what is the difference in the distribution of ACE2 receptor? Is it because the virus gets in there and doesn't replicate at all? Or virus does replicate for some time and then the immune system clicks in and they clear the virus? And what is going on exactly? But even the highest dose, they are clearing the virus within 6 days.
I don't know if it has to do with, your fundamental question is why are they clearing it and not getting sick? I don't know that there's a good understanding of that, whether it's related to the prevalence, location, or abundance of the ACE2 receptor. I also don't know if it is related to the fact that in general, these animals are very healthy. You know, if you look at a lot of the comorbidities associated with SARS-CoV-2, in general, those animals we've either screened out or because of the process of controlled breeding and things like that, we don't have a lot of those comorbidities. So again, that's one of the things that all these models have issues with them. And that's one of the ones out there, but I don't know as it relates to the biology of the actual animal itself. If it's related to the ACE2 receptor or not, I don't know. Let's go to another one on inhaled biologics. It's somewhat the same story. I like this one because I've been working in lung cancer since I was in grad school. I've seen more compounds than I want to describe that I think have great promise in treating lung cancer by inhalation. Fail for business reasons than I want to admit. And I should probably start this one with a story too. Many of you know a scientist named Steve Belinsky. He's one of our senior scientists at Lovelace. He's been working in lung cancer for a better part of 20, 25, or 30 years. When this program came about, the point of this program was Lonza, actually, this group right here, they came to Lovelace. We've been working with them for years. We've been working with them as long as I've been there, and said, no one's doing inhaled antibodies. Why? Is it because of formulations, because of fear? Is it because of lack of efficacy? What is that? They said, let's do an inhaled antibody for lung cancer. We came to bezacizumab. And when I said that to Steve, Steve's immediate reaction, for those of you that know him, and I love Steve, his was, that's a terrible idea. Bezacizumab doesn't work in the clinic. It's not going to work in an animal model. And so our thought was, well, wait a minute. Does bezacizumab not work in the clinic because it's given by injection? Because you're not getting enough local concentration to the site of the tumor? Or does it work because it doesn't work as a drug? And so to me, I said to Steve, you just answered my question. This is a great project to try. Let's see if it actually works. And let's answer that question for this compound. The other thing related to this is, if you look in the literature, there is a lot of people publishing around inhaled biologics. There's not a lot of people doing it. And so part of this was to prove we, Lovelace, and the team we'd worked with on this one, Lonza, could actually generate an aerosol of an antibody delivered in an active form to the lungs of an animal and show efficacy with it. So I kind of just went through this slide quite a bit. That what we wanted to do is be able to show that you could develop an antibody for inhalation. We chose this antibody in this situation because we have a rodent model of non-small cell lung cancer, where we actually take new rats, immunocompromise them, and instill different cell lines, A549, Tau-U6, and a variety of other pretty standard lung cancer cell lines into the lungs of those animals. And we know, one, they're going to get sick. We know how sick they're going to get untreated. And we know the right timeline in the window to be able to treat them. It's about 4 weeks of letting the tumors set up. Then you have about a 4-week window to modulate that disease model. Left untreated at about 8 weeks, the animal's lungs will be so infected with lung tumors, you will euthanize them for morbid indications. So there was some proof that antibodies work in the lungs. Many of you probably know these better than I do. There are lots of antibodies that are given, whether it's systemically, injection, or all kinds of different things for different indications in the lungs. Everything from asthma, infection, lung cancer that we were looking at, and some other models of lung cancer that these are used. So there's proof of concept to say they should work if we can get them there. So our goal with this, the other thing with a lot of lung cancer is you're going in to get these IV therapies. We want to be able to have something somebody could use at home. And there's obviously the expense and compliance that became bigger with other diseases during this previous pandemic. people actually being willing to go in to get treatment for other indications, knowing, my inpatient facility is a hotbed of COVID right now. Do I really want to go in? What can we do to offer our patients something better? How many of you have any background in spray drying? I'll go into this a little bit. So one of the things we wanted was a dry powder of this material. Dry powders generally have some advantages, and I'll go through some of them in a little bit, but with the patients, they're well liked, and there's some appeal to them right now. Let's just leave it at that, both from a patient and from a marketing perspective. One of the best ways to make dry powders is to spray dry, where we're going to take a solution-based formulation of some excipients. In this case, we use trehalose and leucine, and I'll get to them in a little bit, and bezisterimab in that formulation. You spray that mixing with heated, and heated is important, inert gas to evaporate off the moisture, and ideally you get these large porous particles that aerosolize well. But when I teach this session for the Ph.D. students, I stress that heated part. What happens to peptides, proteins, antibodies, and things like that when you heat them? You denature and you ruin their activity. The issue is, and for the engineers or engineering backgrounds, is that when you're using that heated gas and you're evaporating liquid, you have two competing things going on. You have the wet bulb temperature of water that as it evaporates, it rapidly cools down in that local environment. And that heat capacity relative to the heated gas that's mixing. And so our engineering question to this was, which one's going to drive that activity of the antibody? Is it going to be the heated gas? Or is it going to be the cooling effect of that water evaporating at that site right there that'll allow us to have a stable antibody in the back? And we probably found something that worked. So our problem statement was bezisterimab. That's the compound we're going to use. Can we actually deliver that in a non-small cell lung cancer model? There's proof, there's people interested in this. There are, I think, 4 or 5 biosimilars as of today of Avastin out there that are being used. And so our goal was then to be able to use that as an inhalation therapy, not only in that initial window, and I'll get to this in a little bit in the animal model, but also with the idea of maybe this is a take-home therapy. Maybe this becomes a patient has a pretty heavy regimen of chemotherapy or whatever that goes on. Then when they go home, they can go home with an inhaler and take a once-a-week inhaler at home. We tested both of that in the animal model. Dry powders. Your question was actually beautiful from the last section on remdesivir. One of the best things about dry powders is you can take them home and deliver them, and any patient can use them. There's no issue with coordination like there are with some meter-dose inhibitors. But the flip side is, done wrong, dry powders will put a lot of material in the back of the throat, which immediately screens it out from going into the lungs. Our goal was to take a simple, easy device. Because again, Lovelace doesn't own any IP. We want to do great drug development to help people out. To take a commercially available device that you can put a capsule in, fill that capsule with a dry powder material of bezisterimab, and simulate delivering that to animals that would work in a human setting where you get food deposition into a lung. Even with everything that I said about, now we know this patient population has tumors. Likely also, probably going to be something else going on, because a lot of these tumors probably were a part of another disease, maybe a COPD or an emphysema. So you have to make sure you have that right particle size to give them a chance to see efficacy. I mentioned earlier trehalose and leucine. You're going to see those. Go ahead.
So when you have dry powder, I guess you can also give a higher amount or higher concentration.
Yep, I didn't actually show that, but there's a great publication that takes, I think, like 200 different marketed products, and it graphs dose and what device it is. And it's amazing that I'm trying to do this backwards so it's right for you guys. If you look at the dry powder inhalers, they're all high dose and they all group up here. Nebulizers are right here. Meter dose inhalers and soft mist inhalers are all down here. And so when we start with a lot of different people, they're saying, hey, we'd like to develop this, but we want to do a dry powder. They're giving a microgram dose. I'll look at them and say, why? None of the marketed products support that. And probably there's some success and some failure stories in there that you should learn from. That is also one of the really great advantages of the dry powder, is you can give a much, much higher dose. Nebulizers can do that too. But again, where patient compliance is a concern, 15 minutes on a nebulizer, a minute handling my dry powder. I want to believe if a patient has lung cancer, they're going to do either, but let's give them the easier if we can. Good question. So I mentioned trehalose and leucine earlier. Trehalose is effectively a bulky sugar that's just used in the place of API. So if you know what dose of API you want, you can use trehalose to modulate up and down. You can see we actually did that right here in ratios with the API, so you can get the same formulation. Without going too deep in it, leucine was held at 20%. The reason for that is, if you think of the dynamic process of that hot drying air and that liquid feed rate mix, leucine has a lower solubility than just about any other material you're going to put in there. So as that water is evaporating off, leucine concentrates on the surface of the droplet that's being dried. That leucine gives that rough, wrinkly structure that you see on scanning electron micrographs of dry powders. So that leucine at 20% holds that particle, so you can adjust whatever else you want in there. And so you'll see that all over spray drying. Our final formulation we ended up with was the high dose, because it gave us the biggest dosing window that we could give humans. There was our final material. We ended up at about 50 degrees on that outlet temperature. There was a lot of process engineering that went into that. Typically, you'll see that number for other small molecules, whether you're talking about like an albuterol or a fluticasone. You'll see that in the 70 to 100 degrees Celsius. 50 was that right balance for us to see hopefully see activity. I don't want to go into these too much because there's a lot of really, really crazy stuff in here, but what I want you to get out of this is when we evaluated the thermodynamic stability of these materials, we look at when they transition from different physical structures. One of those numbers is a glass transition temperature of 117. This is partially wrong, but the best way to think about it is that tells us if these materials are stored below 117 degrees, we expect their physical stability to be maintained. If you go above that, you have an issue. So in anything we're going to do, we should be in really good shape with these materials. That was our first test. The next thing we looked at was particle size. And the device, useless knowledge, the device that these are tested with, one of those devices, actually created a Lovelace about 70 or 80 years ago by some of the aerosol scientists there. So there's just all sorts of cool stories mixed into this. Think of this, forget these labels. This is the capsule. So this is taking that human inhaler that I showed you and putting it on a device to size fractionate the aerosol, where here's what's retained in the capsule, call it 10 to 15%. These first, then there's the device. These next two, think of them as attempting in a model way, knowing that there's things that's wrong with them, to replicate the human throat. So we can say in this preceptorator and induction port, how much gets stuck in the throat of a human that will be taking this. So for us, this is really, really good to know that we've got a high percentage that makes it down into what would be the lungs of that patient. And remember that particle size that I said. Somewhere between 1 and 3 is going to be ideal. So for us, 2.2 was great. So when we saw this, I started to get excited. When I got really excited was this graph. Like I said, I'm kind of a nerd that way. It's all right. This is, don't even worry about what the assay is. The control is bezisterimab without being spray dried. This is the activity assay of just neat bezisterimab. The blue is after spray drying. This was the one that we were really looking for. As a team, we knew we could make spray dry materials. We know we can deliver these things to animals and humans. Could we do an antibody and not alter its activity? That was really exciting for us. So as soon as we saw that, then we started scheduling the animal study, which is what I'll get to next. Here's a bunch more of the actual potency comparisons on these. Turns out our formulation was really stable, which is great for us to be able to try to find somebody to continue developing this. Let's talk about the animal model. Most of you know cisplatin, very often given in adjunct therapy with different lung cancer agents. So we gave a standard of care, and I want you to look at these doses. Cisplatin held constant at 3 mg per kg. Bezisterimab injection at 15 mg per kg. Inhalation alone at 10-fold lower and inhalation with cisplatin at again tenfold lower. The primary endpoint in this model is lung weight. We tried all kinds of things, and I wish there was a fancy way to do that, but it turns out if you have lots of tumors and lots of things going on in the lung to fight those tumors, lung weight is the best model or the best endpoint in this model. And so that was what we used to really reflect human birth. That graph tells a pretty strong story. Here's your control animals. For reference, an untreated healthy animal rat lung is going to weigh about 1.5 grams. You can see those are some angry lungs. Here's the standard of care, bevacizumab with cisplatin. Bevacizumab alone and in conjunction with cisplatin at a tenfold lower dose. This gives a pretty good hope that we can deliver an inhaled biologic. This would be in that frontline therapy setting. This, and again, these get somewhat confusing, but it's that same graph. This is in, remember I said the model was 4 weeks for tumors to set up, 4 weeks of primary therapy, and then we did some survival studies after that. That's what this is in my mind, and I know it's a model, and I know if you want to poke holes in that, I accept that. Our idea was that's now at-home care, post some intensive therapy. And if you look, cisplatin, it's this is to look at the bevacizumab inhalation with cisplatin. You can see compared to the no treatment, again, you see a reduction even in that later group. And follow the gray line, you see a change from the untreated animals to that treatment and survival. To us, this shows great promise for bevacizumab. Come back to what Steve said. It doesn't work. We agree. It doesn't work when you don't get enough of it to the site of action where it's needed. This program, because this was just proof of concept, this program is now, Lonza is seeing if they can find somebody that wants to actually continue developing this. We have 3 or 4 inhaled lung cancer therapies that are at this same valley of death. They work really, really well. Got some safety data, got some great formulations, but moving them on to somebody that's going to be able to push over that edge to get these into clinical trials, nobody wants to be first. Everybody is afraid to be first. Go ahead, Pete.
You mentioned wanting to do a patient with stage 3 lung cancer. That's not a lung disease. Right? Stage 3 lung cancer, by definition, that really silent volume here outside the lung. So if you want this to work in those patients, you have to get the drug out of them.
Yep.
So how much of this drug is getting out of the lung into the systemic circulation and media silent and all that?
So on this program, we went for the home run right away. Typically, the first thing we would do is what I showed with remdesivir and do a PK study, and we would evaluate blood tissue, brain tissue, maybe some liver, other things like that, along with lung tissue for the IV and the inhaled therapy. So I can answer that question. We didn't do that. One of our other ones, we did that whole gamut and actually showed that the inhalation formulation gives you higher sustainable levels in the brain, in the liver, in the lungs from the inhalation than it does from the IV. And even with that data, find me somebody who gets excited about that enough to help bridge to get it to humans. It's been beating your head against the wall for years. But you're exactly right. We acknowledge and accept that at least early on, any type of inhaled therapy for lung cancer is not going to be first line therapy. No one's going to be willing to do that, by the way. I think, yeah, that was it. So please go ahead.
Actually, control of the say, and the reason I ask this is especially in studying.
We didn't. A lot of us in inhalation try to avoid the fact that you just said that we all know that leucine appears to have some activity in some models. And it's a little scary because as soon as you say that, now you have to treat it as a drug. Now, if you're developing a formulation with leucine in it, now you have to have all sorts of other control arms. So we didn't in this study. Savara was the pharma company that was doing, I don't remember, they're doing an antibiotic that had leucine in it. The whole world was cheering for them. to get leucine as an approved excipient because of your question. Unfortunately, their company failed in phase 3. So we're all looking for who's going to be the first one to do that next, because it's a real question.
Yeah, another question, does it process the area?
I don't know on this one. 2 of the other programs we've done, we did the studies to actually evaluate that. Those were grant funded where we asked those questions, and they did.
Because leucine is also used for epilepsy, right? So I know there's
Yeah.
a lot of
Yeah.
epilepsy colleagues here in the room.
Yeah, so we haven't done that work with this formulation, but it'd be something that'd be super interesting to do.
Did you do anything with parenchymal disease, so-called interstitial lung disease? Because that's all category of diseases where there's some effective treatments, but it's really valid. Antibiotics or immunosuppressants, right? That would seem to be a right target for delivering something directly to the lung, less than possible.
So it depends how broad you want to go in that category. In a lot, if you keep it a little bit narrow in different disease in that space, the animal models are not very good. So any of the fibrotic disease or disease models, whether you talk skin fibrosis or another, they're marginal effects. So a lot of people don't run those pharmacology models or don't put resources involved in those because they're not translatable, they're not predictable. Because of that, we're not doing a lot of work in the pharmacology space in that area. We do more of the toxicology for drugs in that space, but honestly, those studies aren't as cool. They're not as fun to work on. It's a lot of histopath, a lot of clinical chemistry, hematology, looking at more toxicological findings to support safety dose margins. It's not a lot of the pharmacology. Because the pharmacology models aren't very good, we don't do a lot now.
Thank you. 2 questions. We used with the other you call them 10, the hamster model.
Yep.
My question to you, and we can make the hamster become diabetic, even on high-fat diet and stuff like that. Have you tried to put it on high-fat diet and see if by complicating the spectrum, you could have a better dissemination and phenotype expression of the COVID at some point? That was my first question. And since I want to finish, have you tried? You mentioned several times that the pillars of the tonsils are blocking the inhalation, and I agree with you. However, if we tilt the head, those that intubated people, when we tilt the head, you immediately open the back of the pillars just slip on the right side, and it gives you a better access. Have you tried to, especially on the green monkey, have you tried just to lift the mouth of before the inhalation and see if you get a better delivery or a better dissemination?
So 2 questions. In terms of the hamster model for the severity of the disease, it's not cyclosporin. We tried a immunosuppressant model in the hamsters to actually immunosuppress the animals and then see if we could get a more intense phenotype of disease, and it didn't really work. That, in combination with the funding shift away from doing COVID models, we just never pursued it anymore.
There's a drug with diabetes,
Right.
type 1, like
But-
streptozotocin injects. You have a type 1 diabetic, so type 2. Also, if you give a McDonald's sandwich to be
Well, so you can actually give a high-fat diet. They actually have that approved. We can give that.
Absolutely.
But because we never had any funding to do that, we never did that.
Okay.
So you're right. Are there ideas in there? There are. In fact, there's a lot of grants that we've actually been a part of writing to do effectively long COVID models that are things like that. For whatever reason, reviewers don't like them. Is it because the pandemic's gone? Is it because they don't want to think that this might have applicability to the next pandemic? I can't give you one straight answer, but we've never done those models really because of lack of interest. Not because they're not great ideas, just lack of solid funding interest to be able to do that. In terms of positioning the head, that's a really good question. So Chiesi has some really great medical science liaison training programs right now in humans that actually look at everything you're saying. And it turns out that in humans, a neutral head position is the best for aerosols. You go down, which I didn't realize this, you guys probably do that. I were an older person. I'm taking my COPD medication. Oftentimes I'll sit down. Oftentimes I will grab something, whether it be a table or a sink or my end table on my bed, and I'll do my inhalation maneuver like this. That closes the back of the throat. And so myself as a scientist doing non-clinical drug development, I never even think about that. I never even think about that it actually has other changes in the inertial impaction if I look up as a human. Now with animals, if we can get them trained to behave well enough in a chair that their health and welfare is taken care of, that's step number one.
I did a lot of studies.
Yeah.
25 years ago with the gum disease, and you have to deliver
Yeah.
the bacteria, and we immobilize their head a little bit with ketamine and stuff
Yep.
like that. I agree with you. However, you could definitely
Yeah.
move a little bit there.
So when we do these exposures, they're free awake, they're not on ketamine, xylazine,
Okay.
anything.
Fine.
So that's the best we can do. Now, indirectly, we do a little bit of that because the main restraint for safety of the animals and the humans is they're in a chair with a net collar.
Exactly.
And so they're propped up indirectly a little bit to at least hold it uniform from animal to animal and day to day. So at least everything is normalized in the animal model, but in terms of the deposition, yeah, it definitely changes. Other questions?
So with your expertise in aerosolization, so what would be the best platform to develop aerosol vaccines or like vaccines that can be delivered mucosally?
So I work with the Gates Foundation on their TB vaccine. And my gut, and remember, I'm a physical chemist, is that if I could wave a magic wand and put funding wherever I wanted, we would end up with an injection, whether it's sub-Q or IM, as the prime and boosting in a lot of different vaccination settings, whether it's TB, whether it's measles, whether it's SARS-CoV-2, by inhalation. And I would probably do inhalation just because it's a lot more well accepted and you're going to get better coating throughout the upper airways. Again, if I had an infinite amount of resources, I would tweak that statement a little bit. Most intranasal products are delivered on a breath hold. They're delivered into one nostril, and most people hate them because they drip out and they taste bad. We could get a device that does that. Somehow modulates the person's breath so they can't do a crazy fast inhalation, but just a slow, steady intranasal. That is the best one you're going to get because that will cover the primary surfaces of initial challenge, but that will also hit many of the larger airways. I don't know, maybe down to generation 13 to 15 or something like that. So you're getting a good lung dose, but you're also hitting the nose.
Would you advocate for the cannula?
No, just like a tip off of Nasonex or Flonase or one of those, just a very simple, easy one. If I could wave a wand and we could do vaccinations for respiratory indications, prime injection, boost by one of those methods, I think that's the best efficacy you're going to get.
I had an epiphany a few years ago. With the start of COVID, when COVID vaccine, given now, it protects you against severe illness, hospitalization, death. It does not protect the people around you because you're shedding the virus with the lung. And the epiphany that I had was the reason for that was given, actually, it was given, okay, under mucosa, you'd be generating glucose that would get rid of the virus initially in the respiratory tract and then transfer to the bloodstream. So the thing is, what I really love about mucosal vaccine, the respiratory vaccine, I think it's the same thing you're talking about now, is you generate a totally different immune response. Okay, you'd be generating IgA, okay, which is made to do that, okay, and as compared to IgG in the bloodstream, which doesn't do anything. Okay, so in fact, I that's what I would herald. The fact that you stop the disease at the front door rather than at the hospital.
I have no answer, and I agree completely. But and understand, there's all kinds of good and bad reasons out there, and I'll give one bad reason. The story of inhaled insulin. Does anyone know that story about Exubera? If you listen to the story for Exubera was taken off the market because it was a bad product, it was expensive, and it caused lung cancer. And all of those things are accurate if you're the news media. You will find some way to make them accurate. The follow-on device for it was 10 times better. The original device was huge. It was this big and it expanded to this big. You're not carrying it around in your pocket like you do the new devices. Their second generation device was way better. It was expensive. It was a first in class product. No one knew how to price it. And it did cause lung cancer. 4 or 5 people who were previous significant smokers were prescribed a product that had a black box warning to not prescribe to current or former smokers, got lung cancer. Do the math. Did Exubera cause lung cancer, or did smoking for 20 years cause lung cancer? So we're all dealing with that baggage still today of the fact that there was this huge product that Pfizer killed. And the day after Pfizer stopped marketing it, their stock went up. Unfortunately, business is a part of what we do. And so when that's out there, that cuts you off of an ease occasionally. I think we're almost past that, but people still not realizing that the reason for some of those failures were bad decisions. There's another classic one of dihydroergotamine. Dihydroergotamine works wonderfully for migraines. There's a company that failed because remember that graph that I was talking about that said if you have this dose, use this product? Dihydroergotamine is this dose. They force-fit it into a metered dose inhaler that works down here. But dihydroergotamine failed by inhalation. Not because they used the wrong delivery device, but it failed. It's all how the news wants to spin it. So we deal with a lot of that.
It failed also not because of that only.
The biggest
because the migraine, the panel of migraine
right
is huge, and that dihydroergotamine is not applicable to all of those phenotypes.
Right. So MAP Pharmaceuticals was the one that failed with the metered dose inhalers, and their failure was CMC related to dosing of the metered dose inhaler. Satsuma just got bought back in by SNBL on a weird out licensing deal. That's a cool case study. And their program is in phase 3, and I think they're going to get approval. But you're right. You have to better understand what your patient population is and who you're going to market.
I think that if had they targeted the treatment to a certain population of migraine, they would have had.
Yeah. But there's all these baggage of stories of inhalation, things that have failed for all these reasons, and people deal with that.
I thought it kept inhalation failed largely because
it gave a reversible small change in reduction in FEV1, and it was reversible. So when people came off of it, the other thing that really hurt inhaled insulin is right at about the time that product took too long to develop. Right when it was releasing, there were huge improvements in auto injectors. The pain associated with them, the ability to titrate dose.
Yeah. I mean, diabetes is such a prevalent disease.
Yeah.
Even if you pause, let's say, worse than every disease, 4%.
Yeah.
Every emergency room in America is going to be loaded up with patients who are wheezing because they took inhalants.
Yeah. So there were all these things that really the product worked. I mean, Mankind is still marketing it now. They've dealt with the majority of those issues, but if you look at their sales, they're doing kind of this. There's not the hockey stick that you hope for, 3 or 4 years after you get your NDA approved. They have not seen that. Reach out to me if there's anything we can work on.